So horror, vacui, and seeking. Okay, so good time of day, everybody. Now that recording is again in progress, I want to just start with explaining the title of horror, vacui, and the seeming antiquity of root ages. Okay, in fact, this talk is going to be about a recent paper, part of a large collaboration where we dated the bacterial tree of life. But I want to give sort of a conceptual background of what the difficulties were there and how I think we managed to overcome them, at least partially. So about horror, vacui, and the seeming antiquity of root ages, the idea that I want to communicate here is that if one wants to think about molecular dating, in particular about molecular dating at the scale of prokaryotes or the tree of life, where there are very few fossil calibrations, in particular maximum age calibrations, one should think about the idea that nature doesn't like vacuum. Or equivalently, the more modern versions of this, which in physics we phrase as every microstate being equally likely. A gas fills the room evenly, right, because of this principle. And in Bayesian inference, this is reflected in the idea that when we only know some information X, we should assume nothing beyond this information X. That's the maximum entropy principle of Bayesian inference. And in molecular dating, you should really think of molecular ages or molecular age estimates as a gas, okay? To make that a bit more concrete, I want to describe a typical molecular dating analysis, like the one we did for bacteria. So what do we know about dates on the tree of life? I think we know only two things. We know sequences today, genomic sequences today, and differences between them that we can compare. And we have information on fossils, right? Fossilized remains of dinosaurs, or maybe even things like a record of oxygen in the geological record. I'm not going to call all of those fossils. And those are the only two things we know. So some geological information on absolute ages and some modern day DNA sequences. Those are the two things we know. And then we have to construct a model framework which sort of connects these pieces of information back to a data tree, which is our goal, which is what we want to infer. This typically involves things like a substitution model, amino acid substitution model at the scales I've been talking about, and models of fossilization, which I put with a dash box here because we don't really have very sophisticated models of fossilization, and really just have minimum and maximum age constraints at these hundreds of millions of years or billions of years timescales. But to give a more concrete idea, I'm going to set up a simulation where I'm going to sort of think in a forward fashion about this framework. The idea being that I take as known my data tree. It's going to be this nice little toy tree with maybe 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 leaves. And these dates I know, and I'm going to go forward. And what I'm going to do is I'm going to introduce some rate variation. So the molecular rate, at the rate at which substitutions accumulate, will actually be reflected on this tree or generated along this tree. And that's how I get my sequence alignment coming from my simulation. I'm also going to take information on absolute ages through some very simple model of fossilization, where I will take and introduce into my dating analysis some minimum and maximum age constraints from the actual tree or based on the actual tree. And I will know the actual ages. Okay? Because in molecular dating, at these timescales, that's really the only way to represent absolute age information is to have minimum age constraints based on the presence of a particular fossil. For instance, I have evidence of oxygenic photosynthesis at maybe 2.93 billion years ago. That would be interpreted as a minimum age constraint on crown group, on stem group, photosynthetic cyanobacteria. Okay, and that's sort of easy, at least relatively easy, compared to maximum age calibrations, because maximum age calibrations require evidence of absence. There's actually fairly few of these. A classic example is embryophytes, so land plants which produce spores. Spores fossilize very well, and in the geological record, at some point, spores disappear, but the strata in which they should have been preserved where they're there, is still there, like river basins, fossilized river basins. Okay, so Node calibrations come in two forms: minima, of which there are relatively more, and maxima, of which there are relatively fewer, because they're based on evidence for absence, not evidence for presence. Okay, and that's what I'm going to play with. One extra thing we must also mention is that there's a typical shortcut in molecular dating analyses, is that we don't just take as known as data, sequences and fossils, but we take a shortcut and instead of inferring the tree from this entire big model, including the substitution model and minimum maximum age constraints, we infer the rooted tree with substitutions per site branch lengths, so these non-time-like branch lengths, and keep that fixed. So I'm going to do that too here. So I hope that was clear, but maybe I'll try to make it even clearer by this explicit example. So here I have my true data tree. Node 0, node 1, node 2. Here I have the ages for node 1, node 0, the root, node 8, here the other sister group. Black lines are the true ages here. But here I make a simulation where I do a relaxed molecular clock estimate, only taking the fossil information in the very minimal form of having a single root maxima here, the dashed line here. Root is no older than 1,000 units, okay? And I take the rooted tree, but no sequence divergence information. So this is also called the effective prior, which is, I don't think it's a very fortunate name because there's a lot of information here. The fossil information is included as data, and the tree topology is included as data. But so what happens? So what happens is you can see that molecular age estimates are a guess, in that in particular, my estimate of the root age fills uniformly the available timeline. The root could be 1,000 units old, 800, 600, or maybe 100 units old. There is some non-uniform structure as I go lower in the tree to node 1 and 8. You see there's a linear sort of distribution here. That's because 1 must be younger than 0, the root, right? So that's sort of what you have from the uniform distribution. You can actually mathematically calculate pretty easily this distribution of 4, 1. And if you look below 1, there should be node 2, which has this quadratic shape, again coming from 1 is younger than 0 due to the constraints of the topology, and is the same for 2, and so on. Okay, so but what happens if I now add sequence information, here the darker green, I get, well, some more resolution. There is some interactions between the nodes due to the branch length information coming from the sequence, but I get, of course, very poorly resolved and inaccurate. Well, at least it's not very inaccurate, the true ages are within the confidence intervals, but it's very poorly resolved. So what can I do? Well, I can put a tighter bound on the root, both a minima and a maxima, which are pretty close to the true ages. So if I have a tight root maximum minimum combination, then the effective prior will still be pretty poorly resolved. But if I add in the sequence information, I now get actually pretty good ages. Okay? Unfortunately, this is not the case when we're dating practically any tree, but in particular if you're dating bacteria or the tree of life or deep prokaryotic pieces of the tree of life. Okay? We can go further and add some more minima here in dashed and maxima there with the continuous line. And I can see I get increasing resolution and accuracy. Relaxed molecular clock methods do work if you give them enough information, even if you have great variation. Okay, but what happens if, as in real life, I don't have information on when the last bacterial common ancestor or the last universal common ancestor may have lived? What if I just remove these root minima and maxima? What I get is that molecular age estimates, remember, are a guess. They fill available possibilities for age estimates, and the root becomes very old, as do many nodes, deep nodes close to it. So I get an estimate which is practically as old as the maximum bound that I put on the root. And this is arguably often the case. For instance, as you'll see in the bacterial dating estimate, or when trying to look at the age of Luca, we essentially take the age of the Earth as the maximum bound, which is, I would argue, a loose root maximum, which ends up being an informative root maximum, but it's not informative in a positive sense. It's informative in the sense that molecular age estimates you think of as a gas, and age estimates filling all available space. So a loose maximum on the root, if there are only shallow maxima on the other nodes, much closer to the present, can be very informative, can have a very large influence on the final outcome. Okay? If, however, we add more maxima shallower in the tree, I add an additional maxima here on node 9, then I can observe that the root maxima is no longer informative, which is, I would argue, typically a good thing, and I actually get these two maxima an accurate and pretty well resolved age estimates for each of the nodes. Okay? So this is always what happens, I would argue, in prokaryotic dating. And this was the problem we faced in our attempt to date the bacteria. So what we had as a setup, I'll try to speed through this very big paper for our purposes of looking at the dates. We took about 1,000 bacterial genomes from GTDB, evenly sampled. So GTDB has taxonomic classification for over 100,000 bacterial genomes. We picked 1,000 evenly. And we took about 5,000 gene families, cogs, represented in each of these genomes. You'll see why in a bit. And we also were able to take 4 bacterial calibrations, absolute time calibrations, 10 additional eukaryotic calibrations, because we grafted the mitochondria and the plastid onto the bacterial tree as they result from endosymbiosis, mitochondria and the chloroplast. And we also braced, that is set, for instance, embryophyte bifurcation in the tree has both a mitochondria and a plastid, so you can brace those two nodes to have the exact same age. So we did the best we could, arguably taking a lot of molecular data, 1,000 genomes, the markers from there, and as many calibrations as we could find. So this is what that looks like in terms of the effective prior that I described previously, where I don't look at sequence divergence. I don't tell the molecular clock method about sequence divergence. I just tell it about the fossil minimum maximum age, calibrations, and the rooted tree. Okay, what we can see is that, forgetting about the root for a moment, so here, this is old and this is young. Due to the simulations, I had time going in the other direction. Here, it's a reconstruction explicitly. I have 4.5 billion years here, 2.5 billion years, 500 million years, and present over there. So I can see I only actually have one informative maxima that's anywhere near sufficiently old that it's not just in the last few 100 million years. Okay, this one, it's actually the mitochondrial crowns maxima. So what I can see here is that what I have is a loose root maximum. This is the age of the last bacterial common ancestor, estimated without sequence information for now. And it's completely pushed against that maxima at the age of the Earth. Okay? In fact, we can play around with this, and we played around with this. Let's try to raise the piston in our estimate, going with the gas metaphor. So let's try to artificially, just as a computational experiment, change the root maximum from the moon-forming impact to something lower, 3.8, 4, 4.2, 4.51 billion, that's the moon-forming impact, to something even older, 5, 6, 8, whatever, billion years old, billion years ago. And what we see is that the root age keeps going up because molecular age estimates you can think of as a gas. Okay? So in fact, we have a very bad, very ill-defined estimate of bacterial ages that's very strongly determined by the arguably loose maxima on the root. Okay, so what's the solution? Well, the solution is to find additional maxima. Okay, and that's what the rest of the paper is about. And I'll try to very briefly tell you how we did that. So our idea was to use the great oxygenation event. Okay, so oxygen was very unlikely before 2.4 billion years ago, before the great oxygenation event. So it's clear that oxygenic photosynthesis is significantly older, but there were only very small and very local concentrations of oxygen. That's log partial pressure of oxygen compared to today. So before 2.4 billion years, almost no oxygen. And we're going to interpret that as aerobic nodes in the bacterial tree being unlikely before the GOE. Okay, so we're not going to forbid aerobic nodes before the GOE, but we're going to say that they're unlikely. We're going to put a soft maximum there. There's maybe a strong evidence for an aerobic node. This is the age distribution of an aerobic node. Okay, from fossils and sequences. If fossils and sequences insist that something is old, then this soft maxima is going to allow that. But if there's only sort of loose evidence for a node, loose evidence from fossils and sequence divergence of a particular node in the bacterial tree predating the GOE, then the soft maximum is going to push down on that. Okay, so that's what we did. To do that, we need to, in the tree, for internal nodes, come up with predictions for, is it an aerobic organism or is it an anaerobic organism? So we did that with a combination of machine learning and so-called phylogenetic reconciliations, where we took these 5,000 cogs, reconstructed their gene trees, and then reconciled them with the species tree, that is, explained each gene tree as a result of duplications, transfers, and losses along the species tree, which allowed us to estimate the gene content of ancestral nodes. And from this gene content, we predicted using extant labels of aerobic and anaerobic, whether that ancestral node was aerobic or anaerobic. And turns out one can do this very accurately. But of course, that's on present-day data. So how do we try to make sure we're generalizing here across evolutionary distances? Well, what we tested was we trained our model on, for instance, a given group of phyla, bacterial phyla, and then tested the predictions on a group of phyla which we hid from the model. And we could show we get very accurate results. Similarly, we looked at whether if maybe 50% of my genes are missing due to incomplete reconstructions, or there are some false positives, and we could come up with ways to train this model to be robust to both large evolution distances and missing data and inaccuracies in the data. So what we ended up with was this tree of bacteria, where the colors, red is aerobic, blue is anaerobic, and putting that together with the fossil calibrations and the relaxed molecular clock, we could date the tree. But of course, this then raised the question of, are we just getting back what we put in? We're assuming nodes are younger than the GOE. So we get nodes, aerobic nodes, younger than the GOE. Is there any positive evidence for having a model, having dates which are more accurate than dates without the soft maxima and the GOE? So we could show that, in fact, if we use the duplication transfer loss model, then transfers, the likelihood of transfers, which depend on dates, because transfers, there's this example I have to quickly go through. Here's a gene tree. This is a reconciliation of that gene tree with the species tree. I draw this gene tree into that species tree. You see the black part is simple. It's just completely vertical evolution. The red part is a problem. C2, D2 are genes in genomes C and D, but they're in the wrong place in the gene tree. So that's what a gene transfer looks like in the gene tree, this little red subtree. And there's one transfer from this branch leading to B to above C and D. It's one transfer, at least, if you have the ancestor of CD bifurcating after the ancestor AB. Okay? Then it's one transfer. If you change the order and time of those, it turns out it's two transfers. And you can translate this into a probabilistic argument to show that ages of nodes, relative node ages in a dated tree, are reflected in the likelihood of transfers. And what we find is that the likelihood of this model, which includes transfers, increases as we add information. If we only have the root maximum, we get the worst likelihood, we add fossils. Then the GOE with different models, the most accurate model, we get the most likely condition. So we do believe we have better age estimates. And we can also show that we solve the problem of the informative root. So the loose root maximum calibration on the bacterial common ancestor has the moon-forming impact. It becomes non-informative. I raise the maximum, but the maximum age estimate doesn't change. So we believe we have a geological timescale for bacteria that's now independent of the root maxima by using the rate oxygenation event as a soft maxima. And interestingly, this led to the unexpected discovery that several aerobic transitions to aerobic lifestyle, in fact, predated the GOE. And the oldest one of these, at least on the phylogeny of extant organisms, was in the ancestor of oxygenic cyanobacteria. But I don't have time to talk about that. So thank you very much. And I also want to advertise a workshop we're having here at OIST in April. And thanks. I hope I didn't go too much over time.
Thank you very much. That was very interesting. I think quite a different approach from what I've seen other microbial ecologists do. Everyone, please ask your questions in the chat. I will moderate Gergely again for giving this presentation. And we will move on to our second speaker, Dr. Kai Tong, if you can start sharing your screen. He is a postdoctoral researcher at Boston University Biological Design Center, and his work is on experimental evolution and synthetic biology of multicellular life. The title of the talk is Origin of Cell Differentiation and Whole Genome Duplication in the Multicellularity Long-Term Evolution Experiment. Please go ahead whenever you're ready.
Okay, sounds good. Can you hear me and can you see my slides?
Yes, looks good.
Okay, awesome. Okay, thank you so much for the invitation, and I'm very excited to share the work that I did during my PhD on the origin of cell differentiation and whole genome duplication in the multicellular long-term evolution experiments that was pioneered in William Radcliffe's lab at Georgia Tech. So the origin of multicellular organisms from unicellular ancestors is considered as one of the major evolutionary transitions in the history of life. It really paves the way for the emergence of truly complex life forms on the Earth. And it has also occurred multiple times across the tree of life. And some multicellular lineages remains quite simple with simple morphologies and with very few cell types, like these colonial choanoflagellates or these filamentous bacteria, while some other multicellular lineages evolved to become really complex, like in animals, plants, and fungi, where they have very complex, like cell-cell communication, developmental programs, very diverse cell types. So one question that has been intriguing evolutionary biologists for a long time is how do multicellular organisms originate and evolve to become more complex? It is actually kind of challenging to study these kind of questions because we lack an experimental system to directly observe and examine like how multicellular systems evolve in real time. So in our lab, we try to circumvent this constraint by evolving novel multicellular organisms in the lab from existing unicellular species. But I have to emphasize at the beginning that with this kind of experimental evolution, we're not trying to recapitulate how specific multicellular lineages evolved in history. Instead, we're trying to use experimental evolution, this very powerful strategy, to study how multicellularity can evolve in principle. And we can use this kind of controlled experimental system to study how different ecological factors or genetic factors can impact multicellular evolution. And hopefully, in the end, we would be able to extract some common principles about how multicellularity evolved. So in our lab, we use the unicellular Baker seeds, Saccharomyces cerevisiae, and we employed a very simple but powerful strategy to select for novel multicellularity named sampling selection. So basically, every day we just let the culture settle on the bench and then just transfer the bottom portion and grow it for another day. So because you would imagine the larger you are, the faster you will settle, and more likely you will end up in the bottom portion and get transferred. So this essentially select for larger size. And within weeks of this sampling selection, we observed the emergence of snowflake multicellular clusters And this is the result of a single loss of function mutation in the ACE2 gene, which prevents cell-cell separation during cell division. This also leads to a very simple emergent multicell life cycle, where the clusters will just grow clonally, but as the cells grow in divides, the cell-cell packing stress will just build up in the cluster and eventually leading to the fragmentation of these clusters, which is exactly how these simple multicell organisms reproduce. And a previous postdoc and now a research faculty in our lab, also postdocs, really tried to push this system to its limits and pioneers the multicellularity long-term evolution experiments. So he didn't start with the single cell as the ancestor, but instead he started with the multicellular ancestor with the ACE2 gene deletion from the unicellular strain. And because this experiment was initially designed to study the role of oxygen in multicellular evolution, he set up 3 metabolic conditions: mixotrophic, anaerobic, and strictly aerobic. And the mixotrophic condition is just basically just normal Grondy yeasts growing in glucose media. And for the anaerobic condition, he isolated a spontaneous protein mutant from the Grondy ancestor, which basically spontaneously lose functional mitochondria and can no longer respire, even in the presence of oxygen. So the anaerobic condition is basically this petite ancestor growing in glucose media. As for the strictly aerobic condition, he was growing the Grondy ancestor in glycerol media because yeast cannot ferment glycerol. And then he subjected 5 replica populations for each of these 3 metabolic conditions to daily sample selection. And he has been running this experiment continuously for over 2,000 days up to now, corresponding to roughly 10,000 generations. In response to this daily selection for larger size, we indeed see that across all the replica populations, the clusters evolved to become larger. But most strikingly, we observed that in all the strictly anaerobic conditions, all the replica populations evolved to become macroscopic. So you can even see them through your naked eye. But I have to emphasize that these are not many small clusters just sticking together. They're still growing clonally. The reason why the mixotrophic and aerobic conditions are not growing large is just because although they can use oxygen, but oxygen can not diffuse very deep into the clusters. So the inner cells are not really using the oxygen to grow fast. So this essentially slows down the growth rates of these larger clusters, posing an upper limit to how these clusters can grow up. But this constraint is not present for the anaerobic conditions, so they can just reach macroscopic size over 600 days of evolution. And we find that this increase of cluster size can be largely explained on the cell level, especially the increased cell aspect ratio or the cell length. This is because longer cells can allow the clusters to pack more loosely, slowing down the accumulation of cell-cell packing stress, allow the clusters to reach a bigger size before fragmentation. And over time, as the cells evolve to become even longer, they enter this new regime where the branches somehow just entangle with each other, and this allows the clusters to become really tough, really rigid, and very hard to break apart. And this allows the clusters to become macroscopic. And in my talk, I'm going to focus on 2 emerging themes in the multi-experiment, namely the evolution of cell differentiation and whole genome duplication. So without any doubts, the evolution of cell differentiation or diverse cell types is one of the major and also fundamental evolution innovations in complex multicell lineages. So we were wondering, can we observe the evolution of cell differentiation in the multi-experiments? If that's true, that would be like really awesome. So to study this, I extracted a single clonal isolate from the 6 evolutionary time points from the PA5 population, spread across from T0 to T1000. And then I basically used the single cell RNA sequencing, which is a very powerful technique to review the hidden heterogeneity in multicellular systems. And here, like this kind of high-dimensional gene expression data set is basically projected onto this 2-dimensional space where each dot is a single cell. And the cells or dots with more similar gene expression profiles are just grouped together. So for T0, the ancestors, you can basically just see a single cluster of cells, which may correspond to a single cell state. But starting from day 200, very surprisingly, we start to see the emergence of 2 distinct cell clusters, which might represent the emergence of 2 distinct cell states. And there's one cluster that contains most of the cells, and also a minor cluster that contains much fewer cells. And this pattern is kept in T400 and also T600s. And in and you might be able to tell maybe there could be also like a minor cluster in day 0 as well. As I will show later, that's probably indeed the case. And then in day 800 and day 1000, we can see that it's still keeping this kind of pattern of major cluster and a minor cluster. But somehow this major cluster is also dramatically expanding its heterogeneity and form this new cluster. And we find that this new cluster in T800 and T1000 strongly express those stress response genes. And through the labeling of the stress response score, we also see that in the T0 and T600 is also a small subset of cells that highly express stress genes. In the end, we decided to group all the cells from these 6 genes into 3 different cell states: a major cell state, a minor cell state, and a stress cell state. So what are these cell states and how do they evolve over time? So to answer these questions, we use differential expression analysis and also geo-enrichment analysis to study to identify those biological processes that are upregulated in each cell state in each strain. And this is the results. I know there's a lot of going on here, but at a high level, you can see that for each cell state, there is a set of conserved biological processes that are quite conserved across 1000 days evolution, which might be those defining features of each cell state. And there's also some biological processes that are more specific to each evolutionary time points. So the major cell state is upregulating those ribosomal proteins, which indicates that this cell state is probably just actively growing. And the minor cell state is upregulating those cell wall biogenesis genes, especially glucan synthesis. Glucan is one of the major cell wall components in yeast. And the stress state, of course, is upregulating stress response. But starting from T600s and beyond, they also started to upregulate programmed cell death pathways. And we also find that the T800, T1000s compared to T600s have more stress cells and also higher stress response scores than T600s, which may suggest that the T800, T1000 have a magnification of the programmed cell deaths. So this project is still kind of unpublished and is also in its early stage, but we do have some potential hypotheses about the functions of these different cell states. The major cell state may just be an active growing cell states. And for the minor cell states, through this fluorescent staining for the glucan, the cell wall components, we think that these minor cell states might just be those hub cells with stronger cell wall. And this could allow the cell-cell connections to be strengthened and thus increase the multicellular size. And for the stress cell states, through the probium iodide staining, which stands for the dead cells, we do confirm that T8 and T1000 have much more dead cells than T600s. But why is that? Why do we just have like more dying cells? So I think the reason here is because after the microscopic size evolved, the clusters, of course, the clusters are larger, so they can survive the standard selection better. However, this also poses a new challenge for them because they would be so tough to break apart and reproduce. So we think this higher level of dying cells might allow the clusters to fragment more frequently and thus increase multicellular reproduction. And indeed, we find that T800, T1000 have more smaller clusters than T600s. So in summary for this part, we find that the multi-experiments really provide us with a very unique and powerful system to study how cell differentiation evolve in real time and also over potentially thousands of days of evolution. And there are many ongoing studies right now surrounding like what are the spatial localization of functions or the regulations of these different cell states. So please stay tuned for our future progress. So now I will switch gear to a separate story about whole genome modification and its evolution in small experiments. Unlike the previous cell differentiation project, where we specifically designed experiments to test for the hypothesis about whether cell differentiation evolved, this subsequent project is actually coming from a very surprising discovery when Ozan was analyzing the genomes of these T1000 isolates. Because we started this experiment with diploid ancestors. So you would imagine that the novel mutations that we identified in the T1000 isolates should either have a 50% or 100% of allele frequencies. However, what also found is that most of the novel mutations have allele frequencies at 25% and some at 50 or 75 or 100%. And this is very hard to explain unless they would somehow evolve to become tetraploids. So in order to directly test or measure the ploidy level of these different synthic yeast strains, I developed protocol to basically direct measure the ploidy level of these strains. And we find that the ancestors are, of course, diploids, but tetraploidy has already arose as early as the first 200 days of this 1,000-day evolution experiments, and they have been maintained ever since. And further, when we try to measure the ploidy level of the whole populations at earlier time points, we find that tetraploid has already arose to dominance within the first 50 days and then rose to complete fixation within the first 100 days. So this is very surprising, also in very dramatic contrast to all the previous yeast evolution experiments under various conditions, because we know that in yeasts, the diploids are very genomically unstable. So tetraploidy never arise from diploids in those evolution experiments. And if we engineer tetraploids and set them to evolution experiments, they will revert very quickly to diploids within hundreds of generations, because the tetraploids is fit and has some fitness costs, and it's also intrinsically genomically unstable and can lose chromosomes very rapidly and regress back towards diploids. And this is actually quite similar to how whole genome duplication evolved in nature. Although whole genome duplication or tetraploids occurred quite frequently in nature and has played a very important role across the tree of life. However, the nascent tetraploidy is often considered to be costly, and it's also genomically unstable, and they rarely successfully establishes in a diploid ancestral population. So it raises the question of how can tetraploid initially rise and then persist and also facilitate adaptations. And we find in the multi that tetraploidy convergently arises from diploid ancestors, and tetraploidy is also maintained over thousands of generations. So this actually provides us with a unique polyploidy long-term evolution experiments for directly studying how WGD arises, persists, and drives short-term and long-term adaptations. So to study this, I'm going to skip all the details here, but Vivian Chen and then has two very brilliant undergrad students that I mentored. They helped construct these isogenic diploid and tetraploid snowflake yeast clusters. And we find that the tetraploidy can produce larger and also longer cells under both the mixed trophic and anaerobic conditions. And tetraploid also produce larger clusters under both conditions. And because of this kind of dual effect of tetraploidy on both larger and longer cells, so we also collaborate with our biophysicists in our lab Thomas Day to use biophysical modeling to disentangle these two effects on the increase of cluster size. Indeed, we find that both of these two effects can contribute to larger clusters, however, in mechanistically different ways. So we find that if we just increase the cell volume, so the cell number that can be packed per cluster is not really changing. So the larger cells are basically proportionally increasing the cluster volume. However, when cells become longer, we find that the number of cells per cluster is actually increasing. And this is because the cells can be just packed more loosely, and slowing down the accumulation of cell packing stress allows the cluster to reach a bigger size before fragmentation. So as tetraploidy can create bigger clusters, so we think it should be able to offer immediate fitness benefits under selection for larger size. And that's indeed the case. Under competition assays, under sampling selection, we found that tetraploidy can significantly outperform diploids under both mixed trophic and anaerobic conditions. So this leads us to hypothesize that maybe selection for larger size is the primary force that drives the rise and persistence of tetraploidy despite its intrinsic genomic instability. However, there can be alternative hypotheses. For example, maybe our strains that just somehow tend to become tetraploids. Or maybe there can be some molecular mechanisms that evolved over this 1,000 days of evolution that make the tetraploid genomes more stable. So to test this hypothesis, we did evolution experiments to basically examine the intrinsic tendency of genomic changes in these strains by re-evolving them under minimal selection. This is basically done by streaking the strains on the agar plates and let them grow for 2 days and then into single colonies, and then just pick single colonies and streak it again and grow it again. And this is basically a mutation accumulation experiment. And because the selection of the colonies is completely random, this will just minimize the role of selection in these evolution experiments. And after 50 days of evolution, we find that for these diploid ancestors, there's basically no protein change. But for the T1000 tetraploid isolates, as well as our engineered tetraploid strains, they rapidly underwent protein reduction. So these results confirm that our diploid ancestors has no intrinsic tendency to become tetraploids, while the evolved and engineered tetraploids are indeed still intrinsically unstable. So, and moreover, a previous publication that's in tetraploid unicellular yeasts, they found that after 250 generations of evolution, these tetraploid ancestors can rapidly regress towards diploids. And in the meanwhile, they generate tons of aneuploidy, which can be seen from this very complicated like chromosome copy number profiles. And this aneuploidy can actually provide a new genetic route for adaptations. And indeed, they find that this, for example, this gain of chromosome 13 here can provide some adaptive benefits under the selective condition that they grow this yeast in. But that was how tetraploidy was undergoing ploidy reduction. So what about when tetraploidy was maintained? Can it still generate aneuploidy and use it for adaptations? And we find that in our experiments, while the tetraploidy is maintained, we find that in all the PM isolates, so they are still mostly euploids with almost no chromosome copy number gains and losses. But we observed very extensive aneuploidy in all the PA isolates. So why is that? So it did not escape our notice that kind of in correlation with this kind of evolution of karyotypes, in the PM populations, when we compare their phenotypic evolution with the engineered diploid and tetraploid strains, we find that these engineered tetraploids can basically explain the cluster size increase in the PM populations and also in the cell volume and also in the cell aspect ratio. However, the engineered tetraploidy alone cannot explain the cluster size increase in the PA populations and so as the cell volume and also cell aspect ratio. So could the aneuploidy that is evolved in the PA populations also underpin a region of microscopic size in these PA conditions? To test for this, we utilized an anecdotal observation that we had in the lab. That is, when we streak out these microscopic strains on agar plates, most of these colonies will form these donut shapes. However, there can be some rare colonies that form these more spread out, more smoother or rugged phenotype colony morphologies. And when we inoculate them into liquid media, they can no longer form microscopic clusters. And this actually provides us with very simple strategy to screen for spontaneous loss of microscopic size. And we can use genome sequencing to study if there's any karyotype changes or chromosome copy number gains and losses during this donor to sperm transition. And a graduate students that are previous mentors sent the data did some tremendous amount of work that isolated 25 donor to sperm pairs from all of these microscopic PA isolates. And indeed, we find that all these donor to sperm transitions are associated with dramatic decrease of cluster size, cell volume, and also cell aspect ratio. So after we do the genome sequencing for all these isolates and characterize their karyotypes, we indeed find that 24 out of 25 of these donor to spread transitions have karyotype changes, and most are just changing by 1 to 2 chromosomes. And moreover, we find that despite the numerous ways that they can change their karyotypes, the 6 pairs of these donor to spread transitions have convergent karyotype changes. Moreover, there are also convergent changes on the chromosome level, especially the loss of chromosome 8 and also chromosome 14 over here. And more strikingly, we find that all these chromosome copy number changes, except for one, are just changing in exactly the same direction during this loss of chromosome of microscopic size. And even more striking than that, when we compare the direction of chromosome copy number changes in the gain of microscopic size in the evolution of the multi experiments versus during the loss of microscopic size in these donor to sperm transitions, we find that the direction is exactly opposite to each other. So that is basically the strongest evidence for showing that aneuploidy or chromosome copy number changes is indeed playing a very important role in the origin of microscopic size in the multi experiments. So in summary, we find that this multi-experiments is not only the longest running multicellularity long-term evolution experiments, but now also the longest running polyploidy long-term evolution experiments. It really allows us to study how WGD arises, persists, and also facilitate adaptations. More specifically, we find that in the the diploid ancestors arise to become tetraploids, which has been maintained over thousands of days of evolution. And we also find that this tetraploidy can confer immediate phenotypic effects by generating larger and longer cells that can yield larger clusters. And this provides immediate phenotypic fitness benefits under selection of larger size, driving both the rise and long-term persistence of tetraploidy. Moreover, the tetraploidy can not only confer short-term adaptive benefits, but it can also facilitates longer-term adaptation by providing a novel genetic route through the generation of aneuploidy that can create larger and longer cells that eventually drives the rise of microscopic clusters in all the PA populations. And when we remove the unselected pressure, we can find that the tetraploidy can rapidly underwent ploidy reduction towards diploids. And although we know that the real world scenarios will be much more complicated than this, but this does provide us with very simplistic model system to study how polyploids arise and evolve. It's kind of mimicking this kind of polyploidy hop that people have been discussing in the fields about how diploids arise to become polyploids and eventually drop towards diploids. So I know I'm kind of running out of time, but just want to use this slide to thank all the everyone in Radcliffe Lab and also in Sri Kanga's lab in Finland for contributing to all these very fascinating projects. And with all that, I would like to just end this talk, and I'm happy to take any questions. Thank you.